Okay, Monika Richardson, the recipient of the MSSF Harry Tiers Scholarship, joins us tonight to present her talk, The Ectomycorrhizal Fungal Communities of Three Pinus Species on the North Coast of California. Monika grew up on the peninsula and developed a love for the California coast and its ecosystems. She did her undergrad work in biology at Cal Poly, and after working in the field for 7 years, is currently working on her master's degree at Humboldt State. Studying plants led to her interest and now obsession with mushrooms. Someday she hopes to teach at a community college. Please welcome Monika Richardson.
All right.
Okay. Okay. Thanks everybody for coming. And today I'm going to talk mostly about my research, but I'm definitely going to start off by explaining what that first word means. I am a graduate student at Cal Poly Humboldt, which is up here in true Northern California.
Monika, there's just one thing about the screen for Zoom people. Help you with that.
Oh, yeah?
Do it.
Oh,
Just on the computer now. Okay. Says it's sharing.
Oh.
Let's try that. Can you see it now? Okay.
Sweet.
Yes.
Okay. So, this is where I study. This study takes place on the unceded territory of the Wiyot people and the southern territory of the Yurok people in what is now called Humboldt. And so this is a bishop pine forest, and that is a bishop pine associate, Cortinarius turbanus. And we'll get into that, what any of that means. So first off, the word ectomycorrhizal, many syllables, kind of a mouthful, but we can break it down. And basically, ecto meaning outside, myco meaning fungi, and rhiza meaning root, just means a root association between fungi and plants, where the mycelium or hyphocores of the fungi wraps around the cells of the plant. Ectomycorrhizal symbiosis is only one type of symbiosis. And for the vast majority of plants actually do a different type of symbiosis, and that's called arbuscular or endomycorrhizal symbiosis. And 80% of terrestrial plant species do this symbiosis, really common in grasslands and tropical environments. And the fungi that participate in this association are microfungi, and these associations are super ephemeral. So they don't really last very long, but they are super important because so many plant species use those partnerships in order to gain nutrients from the soil. There's also ericoid mycorrhizal associations, or orchid and arbutoid. But today, we're going to talk about ectomycorrhizal symbiosis. I think that'll be more interesting for a lot of you because the fungi that participate in ectomycorrhizal symbiosis, kind of the honky boys and the big players that a lot of us like to collect and eat. And so this is also just a demonstration of the vast morphological diversity of these root tips that are surrounded by mycelium of the fungi, which we will get into a little bit more. Okay, so ectomycorrhizal symbiosis is only done by the higher fungi, and this is fungi in Basidiomycota and Ascomycota. And so within this phylogenetic tree of fungi, I have highlighted the classes where ectomycorrhizal fungi exist. So it's really not a lot. As we know, mushrooms are just kind of a small, primarily in the Agaricomycetes class of fungi. Fungi contain a huge diversity of organisms, most of them being pretty microscopic. The arbuscular fungi that I was talking about earlier that do the majority of symbiosis with terrestrial plants, this is them. This is Glomerulomycota. And it's a super small fungi, pretty boring, not studying that. But it's important, so I included the picture. But what I'm interested in is I'm interested in these classes of fungi that contain our ectomycorrhizal mushrooms. So in agaricomycetes, you have boletes, you have chanterelles, you have pretty much the mushrooms that we know and love. And pizizomycetes and ascomycota, we have morels or cup fungi. And then we have weird truffle-like things in the urocheomycetes, dithidiomycetes, and leosheomycetes. This graphic got a little weird, but basically what I wanted to also talk about was how basidiomycota and ascomycota, the higher fungi, are also referred to as dikaryon. And the dikaryon state is super unique to these two fungi. And what happens is you have two mating types, right? So you have one different type and another type, and they're monokaryon. But those two mating types fuse, they undergo plasmogamy, which means their cells fuse, their nuclei do not fuse. They have cells with two different nuclei that have not fused, that will fuse later on when they're going to undergo meiosis and produce spores, but they maintain this dikaryotic state, which is really unique and is hypothesized to maybe confer some sort of phenotypic plasticity or higher rates of mutation that allows these fungi to actually be super adaptable. So it's a really unique aspect of their life history. On this side, I just wanted to show morphological diversity of the ectomycorrhizal fungi and their corresponding ectomycorrhizal root types. So, when did this begin? Well, it began a long time ago. A lot of people, not just me, think that mycorrhiza or fungal associations with plants facilitated the movement from an aquatic environment onto land. So it's kind of a big deal. And that happened way long ago in the Ordovician era, or period, excuse me, over 440 million years ago. So just conceptually thinking about that, the fungi have always played a role in a plant's ability to, or at least dictated where they're able to grow, has like crazy implications for why fungi have been so fundamental in how our Earth has, and how the like biology of our Earth and the ecosystems found on our planet are formed, or at least enabled by fungi. Ectomycorrhizal symbiosis in particular developed later. Gymnosperms didn't develop until the Triassic period, and we have early flowering plants in the Cretaceous, which is when we presume that ectomycorrhizal associations began. And the oldest living fossil of Ectomycorrhiza is from 50 million years ago. And in that fossil, you have a pinaceous conifer associated with a sister taxa to Rhizopogon or Swillis, for those of you who are familiar with those taxa. And so it is thought that ectomycorrhizal symbiosis probably was happening a lot longer than that fossil was dated to, but that is the oldest that we can say. Okay, so what is ectomycorrhizal symbiosis, or ECM, is what I write in my paper. It is a mutualistic association between the plant host and the microbiome or the fungi. And mutualism in this case is for sure a spectrum. In some cases, in some forest ecosystems, it's not actually fair. It's like the young saplings in a forest ecosystem that are tapped into the mycelial web will actually have a way higher carbon output than compared to what they're getting back. So when we say it's a mutualism, that's definitely a spectrum. And that spectrum can range to like parasitism. However, we also call them partners. We call it mutualistic. And what is happening is the mycelium of the fungus is wrapping around those fine root tips. It's, again, ecto. It's not going into the plant cells, but it's going into the epidermal layer. And the hypha is creating the structure, this latticework structure called a Hardig net. And that Hardig net that's wrapped around the cells in the epidermal layer is where nutrient transfer is taking place. And the nutrients that are being transferred from the fungus mycelium is in the soil. It's mining for soil nutrients. It's mining for minerals, specifically nitrogen and phosphorus. It's taking water from the soil and it's transporting that into the roots. And in return, it's receiving carbon in the form of photosynthates. So when a tree photosynthesizes, some of its output is, cellulose or these highly complex sugar polymers, which really are just carbon. And this is advantageous for the fungi for a number of reasons. If the fungi was just a saprotrophic fungi, which there are many, it would be in competition with other saprotrophic fungi in the soil for carbon. By partnering with a tree host, it has a direct line of carbon, which it needs. It cannot live without the carbon. The carbon allows it to be a building block for it to continue to grow throughout the soil. And so both species or both members of each kingdom do benefit, but it's only a small group of trees, and they are predominantly tree species, that are able to form this symbiosis. However, the trees that are able to be ectomycorrhizal or can associate in this ectomycorrhizal way, they are the dominant trees in temperate and boreal regions. And here we are. And that's a pretty high proportion of ectomycorrhizal plant species. So despite the fact that only around 3% of terrestrial plant species form ectomycorrhizal symbiosis, they are major players. And you'll notice in the tropics, in the region here, a much more highly productive area with higher productivity, more available organic matter, there is not more of an advantage to be associated with ectomycorrhizal fungi. It's in the areas of the boreal and temperate regions where the soil is super nutrient poor. And there's harsher biotic conditions that partnering with these fungi is it's advantageous for the tree, and it allows it to proliferate in this way that we can see here. And this is from a really interesting paper from the Kabir P Lab at Stanford. Kabir is on my thesis committee, and so he's been a huge help because he is a total ectomycorrhizal guy and studying even their consequences in terms of global warming and carbon sequestration. Also, feel free to interrupt whenever with a question. Keep this two-way. But we're going to talk about ectomycorrhizal symbiosis a little bit more until we're so tired of it, and then we'll kind of change gears. But this symbiosis is not just really cool, it's actually super fundamental to a lot of ecological processes on Earth, including carbon sequestration. But the dynamics of this is that it really does create this mycelial network in the soil. So a lot of people are like, oh, the soil web, and it's this new, really cool idea that there's this pulsing neural network of life under the soil where everything's talking to each other. And I think we could go there, but it also is just basically networks of this fungi creating thick hyphal cords called rhizomorphs that nutrients are able to transfer to. And so a fungi might be connecting all of these tree species together through its mycelium. And this creates pathways for nutrients, for waters, for carbohydrates or carbon to transfer from one individual to another. One host tree can be associated with many to hundreds of different species of fungi and vice versa. But there are cases where one species of fungi can only partner with one species of tree. And that doesn't mean one individual. It could still partner with a number of individuals, but it has to be the same species. I was reading this paper last week that was crazy. It was talking about how trees that have ectomycorrhiza on their root or that are engaged in the symbiosis, their hormone signaling in response to abiotic changes is significantly different. They have less up and downs. They have less rapid hormone signaling that results in physiological changes, and that having these ectomycorrhizal associations is almost a mediator for abiotic stressors, which is pretty cool. And then finally, I just wanted to mention that we're all super into sequestering carbon as we emit so much. And really, we think about trees, right, as being these carbon sinks. So we give carbon credits. which is like weird fake money, to organizations to not cut down trees, and that's great. But we don't understand that a lot of that carbon that the tree is sequestering from the atmospheric CO2 is actually being transferred to their fungal counterparts. And a lot of the trees that are sequestering carbon are in these boreal and temperate regions where there's thick forests of conifers. And so this is a really cool graphic from a recent paper that came out, Hawkins et al., ton of authors. Meldon Sheldrake was on that paper, and he wrote Entangled Life, if any of you guys wrote that. Pretty accessible book on fungi. But it dives into how ectomycorrhizal symbiosis in particular is responsible for sequestering up to 50% of a plant partner's primary productivity. And that's sequestering it below ground. And when it is below ground, those fungi are trading with bacteria. And it's staying under, some of it's being respired, but a lot of it is staying sequestered. And another study that came from BOSO 2020 showed that the soil microbiome or the fungi that are present, the different communities of fungi that are present have different effects on how much carbon is being sequestered and on tree growth itself. So, okay, cool. What we just ended on, we're all super comfortable with ectomycorrhizal symbiosis. Every time I say it, no more questions. Everybody's just gonna nod and they're gonna know what it is. And we ended with knowing that the community composition of ectomycorrhizal fungi can have different influences on ecosystem services within a forest. But what are actual influences on the fungal species that are present in a forest? Well, there has been a lot of work done recently, a medium amount of work done recently on these influences, primarily biogeography. The same species or the same type of tree, different forest stands that are super far apart, maybe, that are across the country will have significantly different fungal communities. Host tree will also confer different fungal species that it's associated with. Other factors on which fungi are present include climate, soil nutrient content, and even interspecific competition between different competing fungal ectomycorrhizal fungi. So it's a developing field, and it's important to know the influences on what fungi are present because they do have, there are repercussions for carbon sequestration and for tree growth. Okay, cool. Okay, good. So we all know, we all feel comfortable with that. Right now, I'm going to start diving in to where I live and kind of also where you guys live. And the ectomycorrhizal players that are abound, because really we might not know it, but we are actually surrounded by these amazing symbiotic organisms. Up where I live, the big ectomycorrhizal tree species are Sitka spruce, which goes from Sitka, Alaska, all the way down to Fort Bragg, tan oak, of which there's a ton here. We have Doug fir, grand fir, all of the oaks, coast live oak, white oak, black oak are ectomycorrhizal, and all of the pines are ectomycorrhizal. In terms of fungi that form the symbiosis, we have Swillus, Boletus, Cantharellus, Critiformans. We have Portinarius, Lactarius, Matsutake, Black Trumpet, and of course, Amanita. So these species that are pretty familiar to a lot of us are super yummy or super beautiful, but also super important because they're part of this bigger network. And where I live in Northern California, there haven't really been a lot of studies on the communities of fungi present. We have a lot of natural resources up there, and there's a lot of Forest Service land, and there's a lot of organizations working on conservation, but people are only really just starting to realize and to look into the communities of these fungi that are just as important parts of these ecosystems. And so knowing these communities helps with restoration and conservation, right? If you're going to restore a forest, you need to know the fungal partners that are partnering with those trees in order to inform outplanting or what you're going to try to conserve in terms of soil environment or host species. Not knowing who these guys are doesn't inform biodiversity, and it hurts our understanding of biodiversity because it's a total partial story if we're only really considering one kingdom. And so studies like this add to our knowledge of biodiversity in an area. And then finally, these are obviously, they play a huge ecological role. And so understanding who they are helps us in our knowledge about the ecology of an area. Okay, so I decided to study pines. And I did that because pines especially are obligate ectomycorrhizal hosts. Pines cannot survive if they don't have their little fungal friends connected to their roots. And so there's kind of an interesting thing going on in Humboldt County where we have two range limits meeting in the middle. So the first pine in this study is Pinus contorta, or shore pine, if you guys are familiar, part of the lodgepole pine group. It grows in these stands within the dunes, like you're in this huge dune complex, and then suddenly there's a hummock, and you're in this like pine forest. And it's beautiful. They're early successional colonizing species of sand dunes. They're really important. They stabilize sand dunes in an area, Humboldt County, that is undergoing the fastest rate of sea level rise. It's very important. And then I'm also studying bishop pine, or Pinus marcata, whose range is after my own heart, just on the coast of Baja, all the way up to Humboldt, in all the areas that I would conceivably live in. It is a really beautiful pine that's undergone a lot of trouble. This guy is the documented decline in Mendocino County, where Western gall rust is killing a lot of individuals. It is a fire-dependent species. This species needs to burn in order to regenerate. Obviously, fire suppression has happened. Its last northernmost stand is right there in Humboldt County, and it falls primarily on industrial timberlands, which is okay, but is also kind of problematic. And then I included Monterey pine in this study because it's invasive everywhere, but endangered in its native range. And I included it because it is the closest related Pinus species to bishop. And so I'm interested in comparing host specificity, and because these taxa are so similar, I would expect they would kind of have a big overlap in ectomycorrhizal species. They also share a native range in Central and Southern California. And then I included shore pine just because of the range overlap with bishop. And I also did this because it's expensive to go other places to study. And in this case, I did not have to go anywhere. All these field sites besides Fort Bragg are within like 20 minutes of my house. And that's cheap. That's cheap. I just pay for gas. And I don't worry about anything else. I also included Mendocino bishop pine in this study, along with Humboldt bishop pine, because those two populations are separated by that little hump, which is the Lost Coast range, yeah, where they don't occur. And so that's a big chunk of change right there. That's a lot of miles. And so by including both those communities, I wanted to assess the influences of biogeography on the fungi that were present. Okay, text-heavy slide, but this is basically what I'm trying to do. I'm trying to characterize, compare, contrast the ectomycorrhizal fungal communities associated with these three different pines and between the two populations of the same species. My questions are this: do the associated fungal assemblages differ significantly between the three different species of pine? Will they be similar between the same species of pine that are really disjunct? And will the two native species that have been co-evolving together for who knows how long have a higher similarity in their community composition as compared to the introduced Monterey pine? And put more simply, who's doing the heavy lifting in these forest ecosystems? Because we really don't know. Yeah.
Have you checked Pinus contorta, both the coastal and the montane one?
Like lodgepole pine?
What's that?
Lodgepole pine?
Yeah.
No, because that's far. But I would love, you know what? I would love for someone to pay me to do that. Yeah. Okay. So we're going to get a little bit more into the nitty-gritty about where these sites are. And so you can see topographically that the timber industry is alive and well in Humboldt County, as it is in a lot of Northern California. And yeah, so from this aerial image, the patchwork that we're seeing right here, that's all green diamond industrial timberland. And it's a very stark comparison to Redwood National Parks that starts right at that line. I worked in forest management for a long time before coming to school, and I planned big forest fuels reduction projects. So I'm not here to talk smack on cutting down trees in the name of restoration and healthy environments. But this is something else. And it's working land, it's working timber. We all live in wooden houses. Hopefully someday there will be another solution, but for the time being, this is kind of how it is. And this is kind of where most bishop pine grows. So one of my sites is on green diamond. I got really lucky and I met a private landowner that has an inlet just bordering green diamond, and I use his private property as a site. And then this is the Southern Yurok Territory, Sumeg State Park, right there at the point, where there's just a few bishop pine left right on the coast. In order to study short pine, I chose 2 sites in the beautiful Lanphier Dunes. It is a restored preserve that has been, all invasive species have been eradicated. And this southern part has actually just been donated to the Wiyot tribe, or given back really to the Wiyot tribe, where I have a 3rd site. And then for Monterey pine, 101 to 299, Smo Cookhouse. Yeah, it's pretty good. 1 site there, and then another site just across from the Arcata Airport. And then finally, to study Mendocino bishop pine, I was lucky enough to get a permit from Mendocino State Parks to survey at Russian Gulch, Jug Handle, and McCarricker, which are all super beautiful spots. Okay, so how does one go about quantifying or basically documenting all of the species of fungi that are associated with forest species or with forest trees? The first part's so great. I started grad school. I'd been working so hard. I came to grad school, and all I was doing was just collecting mushrooms. I was going out, I was picking mushrooms. I was like, wow, everybody's complaining about grad school being so hard. I'm having such a good time. And I would go with my advisor. I was learning so much. I was going to different forest sites. I had gotten all of my permits. I had keys. I had special keys that could unlock gates that nobody else could go to. And I was there to collect the mushrooms. I was eating mushrooms. I was having such a good time. But then reality slapped me in the face, because I realized not only would I just have to collect these guys, I would have to come up with sequences to associate all of the mushrooms that I had morphologically identified with their genetic transcribed region, or called ITS, internal transcribed space region, which is a highly conserved region on the genome. It's about 700, maybe 500 to 700 base pairs long, and it is species specific to fungi pretty much. So I collect these bad boys, I dried them to store them in the fungarium on campus, of which I'm also privileged enough to be reorganizing 50-year-old dried mushroom collection. This non-ventilated room. And so I dry them up to store them in the fungarium, and I take a little piece of hymenophore tissue or gill tissue. I stick that in DNA extract, and then I amplify that region, and I check my results with, oh, not so good, gel electrophoresis. So yeah, this has kind of been a big learning curve. But we're learning how to Sanger sequence mushrooms. Okay, the second part. For those who know me, not my real smile. That is a grimace. That is a pained look. Because the more important part of quantifying these communities, you can collect mushrooms, right? And be like, oh, I found these here. But there's a pine right here, there's a spruce 200 meters away. You don't actually know if that fungi is in symbiosis with that tree. So what you gotta do is you get yourself a little bulb planter, and you take a soil core like 6 inches away from the base of a tree, and you do it on either side. and you make a conglomerate soil collection, basically. You fill the Ziploc bag with the soil cores you took from either side of the tree. And you go through that soil core, and you go through all the roots, you sieve out the dirt, maybe you wash the roots, you wash the dirt from the roots, and you use a dissecting scope with a attached camera and going through every single root. You put all of the different morphotypes of Ectomycorrhizas, and these are the Ectomycorrhizas that we talked about at the beginning. This is the site of symbiosis, with mycelium is wrapping around the fine root and creating this morphologically distinct shared organ. And so I go through each sample. I put all the different morphotypes I see into groups, and then I take representatives from each morphotype, and I put it in a little plate. I weigh it out, has to be 0.1 grams. Throw that sucker into a tube, throw that tube into a negative 80-degree freezer. And then I had big dreams. I was going to do all the sequencing myself. I was going to teach myself how to do high throughput sequencing, next generation sequencing. And then I said, why? Nobody's paying me. And I don't want to be a molecular biologist. I want to be a forest ecologist or like a fungal ecologist. And so I sent those suckers off. And I chose FedEx. And FedEx lost my samples for 24 hours. My first gray, swear to God, first gray hairs that I've ever seen on my head came in the week after that period. I called them. I was calling everybody. Where are my samples? They, oh, maybe they're in Chico. I think they're in Kentucky. They're back in San Francisco. Oh, they're in New York. Nobody knew. And so I was just calling. And then finally the next day, they found them in Virginia. And they put them back on the plane, and they finally got to Long Island, and I got a refund, $300 back in my pocket, and hopefully, hell yeah, hopefully, and so fingers crossed, you know, I was like, okay, maybe everything's okay, and for the most part, it has been okay. And so that was the Ectomycorrhizal root tip collection. And then finally, like I mentioned, soil type can be an influencer on the fungi that are present. Kabir P, who I mentioned earlier, he was a part of a study at the ecological staircase at Jug Handle State Park in Mendocino, and just studying bishop pine that grew on different levels of the terrace, they found significant differences because of where in the terrace that tree was. So you have younger soils, and then you go more oligotropic soils as you go up the terrace. So I couldn't really do this project and say that host tree or biological or biogeographic distance was the influencer if I did not also test soils. And this was a very aesthetically pleasing part of the project that I sent to my art friends to appreciate. Okay. So like I mentioned, I am in my second year, so I have done a lot of this work. Haven't done a ton of data analysis yet, but I do have a lot of pictures that I'm excited to show you guys because I spent so much of my life looking through them. So I don't know if this will mean much to you, but I thought it was just cool to have a pictorial representation of the diversity that I found at each site. So here, Humboldt bishop pine. Here we have just a few of the fungi that I found, and then some of my favorite Ectomycorrhizas that I took pictures of with my little scope. So we have Hydnum, we have Tricholoma, we have Lactarius, Coltricia, Cortinarius, we have Suillus umbenatus, of course, more Tricholoma, and yeah, more Pseudoleucaria. And then over here, we just have a bunch of beautiful root tips. And honestly, by the end of going through soil samples, I was convinced that I was actually more interested in looking at these guys than these guys, because the morphological diversity is like really astounding, and they're so cute. For Mendocino bishop pine, I went twice to collect mushrooms, and I got a few, but I usually went after a storm, so I didn't have my beautiful picture collage, but I do have beautiful pictures of Ectomycorrhizal root tips that I just think are cool because they show the morphological diversity. For shore pine, up where I live, there's a ton of Russula, there's Amanita, there's Rhizopogon, we have Suillus tomentosus, different species of Cortinarius, Thelephora, Hydnellum, Inocybe, and of course, Matsutake. Having a permit to Lamphere Dunes is pretty cool because nobody goes to my spots. I have to just keep renewing that permit so I can keep going to those Matsutake spots. But here are some of the root tips associated with shore pine. And then finally, for my beautiful roadside sites where there are Monterey pine, there's of course Philetus edulus. There's a bunch of Russula, there's Amanita, Swillus pungens, not Amanitas, and various Tricholoma species. And then here are their corresponding root tips. One thing that did happen, unfortunately, is I sent in 48 of those little tubes, of those root tip tubes, and 39 of them amplified, which is great, but 9 of them did not amplify, and 8 of the 9 that did not amplify were from Monterey pine. So at first I was like, oh my God, FedEx, I bet this was you somehow. But I was talking with some people and they were like, you know, all of these sites are roadside sites where people are spraying pesticides, or like road maintenance is probably, you know, putting some weird stuff in the soil. And it could be that either there's like weird chemicals that break down DNA faster, or you're getting these root tips from older Monterey pines that are pretty resinosus. They have a lot of resin in their roots. So maybe that disrupted it. Either way, luckily, 4 is the smallest number you can use for statistical significance, and I have it. So here's another kind of boring, overwhelming slide of all the individual species found at each site. Again, Monterey pine is a little smaller because it's only N equals 4 in terms of sample trees. Mendocino's N equals 11 because 1 didn't amplify, and both Shore and Humboldt Bishop pine N equals 12. So those are, nobody wants to look at that. Okay, so what have I found just from looking through this data so far? Here are the heavy lifters in terms of sequence, or some of the heavy lifters in terms of sequence, how many sequences were found in the samples. There was a ton of Cortinarius species, and there were a ton of Russula species. And this is why I need to, and why I am Sanger sequencing mushrooms. In the data that I got back, I basically, we ran bioinformatics and we compared the OTUs, or those genomic regions, to known genomic regions using the UNITE database, which is a really cool database that's out of England, and it has ITS or even full genome sequencing for ectomycorrhizal fungi. So for a lot of these, it just came back Russula species, Swillus species, because that's all that was in the database. And so what I need to do is I need to, and what I am doing it, it's just slow, is Sanger sequencing the mushrooms that I actually find, so I can compare the site-specific genomes that are found within Russula or Cortinarius to those OTUs that were coming back only to genus. But yeah, so here are some big players. Swillus umbenatus, only found under Bishop and Shore, not found under Monterey. Swillus pungens is found under Monterey, and Swillus pungens is found under Bishop in down in Point Reyes. So what's going on is we're kind of seeing Swillus, which is a really important mycobiome. It's pretty key in ectomycorrhizal symbiosis, especially with pines. We're seeing that umbenatus is kind of the associate up north, whereas pungens, which is in Monterey, is more of an associate down south. So already some discrepancies in who the fungal partners are. But more interestingly was so many of the sequences I got back were from these resupinate crust fungi, crusty guys. These were the players that were doing some of the, like, probably some of the highest proportions of reeds were these fungi. And Tomatella subliliciana, this little guy was in all of my samples. Piloderma was in a ton, Trichospora, Tylospora, but these are these fungi that really don't have a lot of recognition because A, they're not edible, and B, they fruit in the duff, under the soil, under logs, and moist environments where their spores aren't really being aerially dispersed. So you're not really coming across them, but they're so important. Like, they really were on almost all root tips. These guys that you don't really appreciate, or I didn't, are doing the heavy lifting in these environments. And I wanted to talk a little bit, just a little bit, about Tomatella sublilicina, because it is actually kind of a famous fungi. It's found all over the world. This is from gbif.org, and they're pretty heavy in Europe. So that's why it's showing that there's so much representation there. but really this species is found everywhere. And its spores, like I mentioned, are not airily dispersed. Instead, they are dispersed by going through the digestive tract of fungivores and insects who will eat and munch on these crusts and then go and then poop it out, and then that will grow somewhere else. And going through the digestive tract, you'll see that these spores are actually pretty spiky. They're evolved to do this. And some people think that that confers a better ability to germinate, if you will. And so I just want to talk about this because I think that this is what's going on in these ecosystems, that these fungi are being spread by these insects, and they are like widely dispersed because this has been such a successful mechanism. Okay, so I know who is where, I know what's going on, kind of, but now the work ahead is to compare the communities. And I've done it. Just kidding. Just kidding. I actually forgot to separate Bishop into its two different places. It was last week, 8 p.m. I was like, I got to have one slide where I showed that I did a little something and I hadn't used R in a really long time. And so this is what I'm turning in for my thesis. This is just a NMDS, or non-multivariate dimensional. It's an ordination. It's an ordination where basically each dot represents a community of fungi from each sample, and where they're placed on each axis represents how similar they are to each other. So this one is pretty different from this one, and these two are pretty similar. This is why it's unfortunate that I only have 4 Monterey, but also why it's fortunate why I have at least 4 Monterey, because when I run a permanova or statistical analysis on this, I will be able to have some sort of backing behind me. But this is not a final result at all. And a lot of these will change because the communities in some of those are only specified to genus. I still need a Sanger sequence, and I need to add soils and soil characteristics to really compare these communities and on which axis they lie. So here are all the statistics that I am going to do. They're actually not that hard. I realized you just need to know R, which actually is quite hard. And if anybody does know R, let me know. But I really think that I can just buzz through this. And really all I'm looking to do is taking richness estimates, measuring diversity, measuring how similar or dissimilar communities are, showing their similarity using a non-metric multidimensional scaling ordination, testing the significance of that using a permanova, and then I just added this. I just added that. Okay, so like I mentioned, next steps are, here are my OTUs from root tips. I need to compare these sequences to the Sanger sequences that I will get from the basically my PCR products, or what I've amplified. When I'm done doing PCR on all of the mushrooms, I send out a plate to Eurofins, wherever they are, and they do a quick Sanger sequence. And then also, using the very gracious funds from the Mycological Society of San Francisco, I will send off my soils to be tested. Okay, let's bring it all back. Why am I doing this? You might be wondering. This seems like a lot of work. Or also, why are you doing this? I don't understand why you're doing this. But I still believe that this is important because we really, like north of Point Reyes, there haven't been a lot of studies on who these guys are. And like I've been mentioning, they're pretty key in maintaining these forest ecosystems. Biodiversity surveys such as this help us know what we're even trying to conserve, especially shore pine is threatened in its native range. Bishop pine is facing decline in pretty much all of its range. If we want to do outplantings, we should be inoculating seedlings with fungi that we know that they're partnering with. And I hope that this can be kind of a baseline study that informs future studies on how to whip one of these out in 2.5 years or quicker. And then finally, yeah, I think we all know what's going on. Things aren't looking super hot, or they are. Things are looking super hot, and a lot of a lot of forests are predicted to decline, especially in temperate and boreal regions. Yeah, they're climate change, drought, a whole lot of things. And these forests are sustained by ectomycorrhizal fungi, but at the same time, they also sustain the ectomycorrhizal fungi. So we need to understand what's going on now as we are right on the doorstep of like serious ecological catastrophe. It's important to take note of what is here, and maybe it can help us to combat what's going on. Yeah, the associated biogeochemical cycles in pinaceous forests, which we were talking about, like carbon sequestration, will be a really important way to combat climate change. So it's not really just about keeping forests around so that they can sequester the carbon. It's also making sure that we're also thinking of the fungal symbionts so that we can like further conserve and sequester carbon. Okay. I hope this picture means something to you now because it means a lot to me. And I just want to say thank you so much to the Mycological Society of San Francisco for this really substantial award that like dramatically helps me do this research that is not costly. The gel company, my dad's company, that I conveniently timed this degree with my dad's slowly being done with his genetics company that he started 30 plus years ago. And he just has all of this DNA extract and master mix that he doesn't want anymore. He has nowhere to put it. And so my dad had donated a lot of costly supplies like DNA extract or master mix, DNA ladder, that has enabled me to do this Sanger sequencing for a lot cheaper than I would have otherwise done it. And then also thank you to other mycological societies and other sources of funds that have helped this project. Here's evidence that I did some research. Thank you very much. And that's it. Okay. Wow.
So do you have an idea of the overlap between fungi that are on pines and fungi that are on like Ectomycorrhizal or something?
Yeah, between Fagaceae and Pinaceae. There's definitely some overlap, like Amanita muscaria, you'll find pretty much under everything. You will Swillus Swillus is mostly a pine thing. If you with oaks, you definitely have more like Boletus, I've noticed, or not Boletus, the whole Boletales, the order in general. But I haven't quantified that, but I would if somebody paid me. Yeah.
So, in your methodology, you looked at 6 inches away from the trunk of the tree, both sides. I assume part of that was because it was difficult to find pure stands of these trees.
It is difficult to find, not for shore pine and not for Monterey because it's the only thing that's planted, but bishop pine is definitely kind of was difficult. But actually, you would be surprised when you are 6 inches away. There's a ton of roots. I was with Ben, my private landowner, and he was like, "What do you do? Like, let's look more over here." And I was like, "Okay, Ben, we're gonna do this just to show you they were wrong." But you go like 6 feet away, and it's actually a lot harder to find the roots. But when you're at the base of the tree, even in that duff layer, because pines do have so much duff, that's actually the perfect place to find Ectomycorrhiza. And so 6 inches is something that I saw in a lot of Kabir studies, so I replicated it that way. But also I think Kabir figured out that is kind of a good distance to get a pretty high density of Ectomycorrhiza.
So that kind of leads to another subject, and that is succession with fungi, that you find different fungi with young roots than you do with older roots. Do you think you would have found a bigger diversity if you had gone out to the ends of the root tips?
Okay, yeah, like if like early colonizing fungi where the roots are still growing.
Yeah.
I think that's possible. I have read papers that talk about seedlings having different ectomycorrhizal communities than more mature trees. But I think that if it was a more mature tree, the ectomycorrhizal fungi are in the soil, whether it be spores or mycelium, and they can exude chemicals basically to colonize that root. And I think that if a root was colonized over here, there would maybe be a pretty similar fungal assemblage that would also be over here. But if they were seedlings, I wouldn't say that as confidently because of the studies that I've read. I don't know if it's because of chemical properties or like different enzymes that fungi use in order to colonize tree roots. There is more of a discrepancy, but that could be a thing too.
Did you look at successional fungi on a particular tree?
I took all of these cores one time, so I'm really just trying to quantify the community that I'm finding. And this is kind of like standard methodology for doing like a first entry into a forest ecosystem, or yeah, into an ecosystem. Mhm.
What's different about pine trees compared to other trees that they require the symbiosis?
Yeah, a lot of pinaceous conifers, which includes like spruce, are also ectomycorrhizal, but spruce is not as highly colonized as pine. I think pine is so co-evolved with ectomycorrhizal fungi. Like I mentioned, it's like the oldest fossil. I think pine has lost some of its ability to do some of the soil exploration through its roots that other lineages of trees have. So like, that still have. So pines associate with so many fungi, and they are kind of like rich partners in fungi. And I think it's because they're more reliant on getting their soil minerals and nutrients from these fungi. Whereas other trees like spruce, I'm not really sure about any like Fagaceae or oak, still maybe have more of an ability to colonize with their roots. And those trees also could not grow normally without ectomycorrhizal symbionts, but maybe they don't rely on them as heavily because maybe they're not as co-evolved. But I cannot say that with 100% confidence.
What were some of the biggest challenges you faced in your research?
Oh.
Additions of facts.
Yeah. Yeah, well, that's a loaded question, but I'd say I had to go redo a lot of soil cores because at the beginning, part of this study was only choosing host trees that were at least 15 meters away from another ectomycorrhizal tree species. But I learned that the roots actually go pretty far in the soil, and I think it took a really long time to get comfortable recognizing pine roots specifically, and that has just been through like understanding that the cambium layer in a pine root is really red, that when I scratch it with my fingernail, it's going to smell differently than a spruce or fir. And then also, yeah, just getting really familiar with the way that the root looks. So that was having to go and redo a lot of this stuff has been tricky. Yeah. And that's all I'll say. Yeah.
Is there any difference if there's like ferns or other understory plants in the area?
Yes. Salal. You guys know salal kind of grows here? Yeah. Okay. Salal will grow in huge chunks, or I'll get like a chunk of huckleberry. And I'm realizing, or I learned that when you have these like understory plants, whether they be herbaceous grasses or more woody like perennial shrubs, the area, like there's competition for area in the soil for these roots. So when I'm looking in an area like that where I'm needing to dig through salal, I'm going through so many salal roots and way less pine roots because the pine roots can't proliferate as much as they can when they're just in kind of like the understory is just needles. And so totally I realized like, okay, if I'm seeing like thick salal or thick huckleberry, like I'm not even gonna bother because I know it's gonna take me so much work. And that is like root competition in the soil, which exists between all plants. Any questions over there? Anything else? Oh, yeah.
You have a question on Zoom?
Okay, wait. Will you read it?
Yeah. Maybe.
Yeah.
I know Tom Horton was doing something where he was looking at how far away from the tree that there would be enough spores that they would inoculate a seedlings or something like that.
6 miles.
Yeah.
Spores can travel 6 miles. Tom Bruns did that study, or maybe it was for at Monterey or at Point Reyes, yeah. And basically was like had was testing, like had boxes out and would basically like sterile boxes and test how far spores would come. And he was able to trace certain fungal spores from their origin, and it drops off dramatically, but spores can travel up to 6 miles. And so that's like a serious mechanism for dispersal.
What about the rhizomes? Do they travel underground?
Yeah, they do. I don't know how far they travel. They definitely don't travel 6 miles. But they probably travel closer, like maybe 50 meters or more, or somewhere around that. Well, you know what? I just made that up. I don't know.
All right, so is the effect of forest management events on mushroom habitat ever taken into account in official evaluation for permitting or regulatory purposes?
Okay, I think I understand that question. I was actually doing a job interview today. We were talking about rare fungi services surveys that the Forest Service is starting to do. So like they do rare botanical services or rare botanical surveys. Doing that same thing for fungi has just started to happen. And so that's kind of thinking about forest management, because if you find that rare fungi, you're not going to use heavy machinery. When you use buncher fellers or really big heavy machinery to clear a forest, you are compacting the soil in a really dramatic way. And you are prohibiting, when you compact the soil that much, you can be making it hydrophobic and also much harder for mycelium to move through the soil, or it can kind of like destroy the fungal communities that are there. So I was working in forest management and I was creating prescriptions for buncher fellers or feller bunchers, sorry. And I heard about this study. This person came to the resource conservation district I was working at, and they were like, hey, what are you going to do? But it's kind of this really hard thing, because if you pay for all hand felling because you don't want to hurt the fungal community that's there, because you don't want to compact it, you're paying $5,000 to $8,000 an acre to thin an area, whereas you would be paying $1,500 to $2,500 to use feller bunchers. So I think that is growing. That understanding is growing. People are trying to consider inoculating trees before they plant them, but it's kind of just starting, I would say.
Are you aware if forest managers, either preservationists, conservationists, or government actors, have shown any interest in accounting for the role of fungus in forest propagation?
Totally. Oh, wait, does it keep going?
Yep, go ahead.
Okay.
You're good.
I just kind of like I mentioned, like it is just starting. I, in the same interview, which I think I did pretty well, we were talking about this one mycologist was like, hey, we should be careful before we inoculate seedlings because we don't want to spread invasive fungi. And that is true to an extent. A lot of the invasive fungi that I come across are saprotrophic, or they're just litter decomposing fungi. But Amanita muscaria is technically an invasive fungi. And that partners with a lot of stuff. So yeah, maybe consideration. I was hearing from this other mycologist that this new kind of growing field of wanting to inoculate trees before you plant them. And this is a huge thing in Northern California. Like the fires have obviously devastated forest ecosystems, and there is a really big push for replanting, collecting seeds and putting them into a nursery and then bringing them back to sites. So this discussion on like inoculating is kind of what's I'm hearing the most in natural resources, and then also the discussion on not using heavy machinery. And so I think that fungal surveys, like rare fungal surveys, will prevent heavy machinery from being used, or certain treatments, the same way that rare plant surveys do. I think I saw it back there.
So in the microscope images that you shared, I believe that they were of the actual tree roots.
Mm-hmm.
So how were you able to distinguish which of those roots had the EMC on?
Yes.
Like, because I couldn't see them, how could you tell that they had a?
You know what? Great point. I should have just shown a naked root. It's very obvious.
Okay.
A naked root is just like very underwhelming. Those pictures were like, maybe not to you guys, but to me, like they are just so beautiful and shiny, and they have like special colors and like special morphologies. And it might be to the trained eye, which is a good point. I should have shown how a regular root is just like, and it has like, like a little stick coming out. Like there's no like fuzzy little weird guy on it. Yeah.
About that, how did you find? Was it you found the cluster guys? Like, how did you find them, and where were?
Yeah, dude, I didn't even find them. That was just, those were the sequences that I got back from the root tip. So some of those Ectomycorrhizas that I was like, I don't know whose that is. When I ran it through next gen or like a Illumina MySeq, the sequences that came back were those fungi. And so now when I go to those sites, I'm like looking under logs, I'm looking through the needles, and I'm starting to like take collections of those so that I can have site specific like ITS sequences from those guys. But now it's like really changed how I walk around a forest, because obs I'm still trying to eat. I'm looking for the ectomycorrhizal like honkers, but I'm also like, who else is here? And you really realize that like all wet surfaces are covered in fungi. Yep.
So speaking of wet surfaces, did you take all your samples, did you collect them all last year?
I collected them over summer, right before you visited me.
So is it? But so we can say if you just entered this, program this year and there was no rain, like how
Ooh.
would you do?
Totally.
See wet surfaces and stuff, is that
Totally.
obviously impacting it, but like, how is it?
How would that change things?
Yeah.
Well, I actually collected the root tips in the summer because it's easier to collect in dry soil. And also it's never truly dry in Humboldt, like even in the summer. Record high in Arcata is 85 degrees. And that was like an anomaly. There's always like summer fog. And so I didn't want it to be really wet when I went through the soils, because if they were saturated with water, there would be a lot of contaminants, like a lot of water molds. But I was super lucky because last season was this amazing rain year. If you guys maybe remember, like fruiting last year in 2023, wait, what? Yeah, oh, 2023 was amazing. Fall of 2023, we got so much rain. So I did the majority of my mushroom collection then. And I went back and was collecting now in fall 2024. And it was bad. It was weird. Phenology and how mushrooms have been fruiting, at least up north, is pretty off. It's off from how it usually is. Things that are supposed to happen later are happening way earlier. And yeah, so I actually am relying really heavily on the mushrooms that I collected last year. Yeah.
You're sequencing both the mushrooms and the rhizoids.
Yeah, and the root tips to compare. So when I have unknowns in the root tips, I can compare to the mushrooms. And because my advisor was like, I'm not going to be interested in this project if you're only doing soil stuff. And so we went out together.
I saw you had Lauratia Monetary up on your Ectomycorrhizal.
Yeah.
I thought that was just a wood decomposer. Well,
No, I found this amazing paper by Tedersu, who Liho Tedersu is like this ectomycorrhizal superstar. And he just went through all of the orders, and there is one genus it might be Piloderma, in Leoshiomycetes that is ectomycorrhizal. So that's why I included it. But if you email me, I will email you that paper. Yeah.
Does the diversity of the fungal associations confer some sort of protection to the tree against pathogens? And has anyone been qualifying?
Yeah, in one of the earlier slides, this a study that I cited, the microbiome of the soil confers different growth rates of trees and different rates of carbon sequestration. So it also confers like you were kind of mentioning, like resilience against fungal pathogens. I can't say it directly said that increased biodiversity was correlated with increased resiliency, but that does seem to be a general trend in ecology, that biodiverse ecosystems are much better suited in the face of adversity. But yeah, I cannot definitively say yes or no. I think so. Oh yeah. Yeah. But yeah.
Did you have any striking encounters with wildlife?
Yes. Okay. Great question. I'm digging, right? Because sometimes it's like, yeah, I can use the post or the little bulb planner, but a lot of the times I'm using my hands. So many times I'm digging and then I start to hear like, and I'm like, oh no, oh no, oh no. And every time I would switch to a different tree and I would dig and then I'd hear like, and like, I'm like, oh my gosh, it's a killer bee. It's the first killer bee in America. But it's the little carpenter bees. It's the little black bees. They like bury in the soil. But they would really scare me because then and then other than that, no, it's like a lot of frogs, some birds, just a lot of bees. Yeah. Again.
Is there any wildlife that eats?
Yeah. Ants, yes. Oh my gosh. Deer eat them, squirrels eat them. In Australia, actually, ectomycorrhizal fungi are eaten by marsupials. And that's a way of their like spore dispersal. But in these environments, like I would see a lot of russulas that get chewed up, just like destroyed. And I think that was probably a squirrel, or definitely not
Slugs.
slugs. Oh, slugs for days. Slugs. You always find slugs underneath mushrooms. And like I was saying about tomentella, like it's eaten by insects and like other fungivores. But yeah, for sure, animals are eating mushrooms big time. Mhm.
Oh, what about there was a paper that was talking about, I think it was, hygroscopies.
Okay.
They were finding those in the leaf tissue and stem tissue of, organisms, and they were still having the fruit bodies out there. But they
Whoa.
weren't necessarily associating with the roots.
Oh, like were there spores on the leaf, or like it was
Endophytes,
What's that?
endophytes,
bacterial
They were,
endophytes?
they were, well, what was the question?
endophytes.
Yeah, endophytes.
Okay.
And so it's interesting, if you're looking at the roots, you might be missing a whole bunch of fungi that could be associated with the tree
Yeah,
and interacting differently, that's all.
definitely. I guess it would be hard to sequence those areas and not know if you were just getting spore contamination.
Yeah, I think they were chopping them up and they were doing the sequencing of the leaf tissue.
Oh, interesting.
Yeah, it sounds really weird, but
I'm not touching that.
yeah.
Yeah.
In all of your time with your dissecting microscope, did you ever see any like morphological specificity to like looking at one species or with one tree root species, you know, like, oh, I keep seeing this like same type of like sheen in like, which I know you grouped everything together instead. Could you eventually in the future like separate them out morphologically and see if it's like a specific?
Yes. One of my biggest regrets is not Sanger sequencing the root tips, because I like bulk sequence the root tips. So I'm just like throwing them all in and then getting a bunch of stuff back. But I got so many of these like black hairy root tips, and they were just like single little root hair or root tips covered in black, like fuzzy with like emanating hyphae off of it. And I was always like, man, what is this? And now like there's this really cool website that you should check out. It's called Deemy. DEEMY, and it's basically a database of ectomycorrhizal root pictures with a corresponding species. But I was seeing like, I was like, okay, wow, like Bishop pine has a lot of these guys, and I'm not seeing that on Monterey pine. But if I had Sanger sequenced each different like root I had seen, then I could be like, this picture is this species, and that actually would have contributed a lot. But I did not do that, and maybe next time I would. Mm-hmm. Yeah.
I'm curious to know if there, you mentioned that one of the pine species you were studying is fire dependent.
Mhm.
I was curious to know if there are any studies on the fire resilience of different fungal species and how it interacts with that tree.
Yeah, we were talking about this at dinner actually, and how after a fire comes through an ecosystem, it can take times for, or it can take a while for some fungi to start fruiting again. And usually after fire, you will get a lot of ascomycetes fruiting, or that cup fungi, like for instance, like morels, right? Morels follow fire. But more than just morels, a lot of cup fungi that belong to Ascomycota are fire followers. So you actually do get like a different assemblage or a different phenology of fruiting post-fire. And there are studies on that. And Bishop pine, there are studies on that on Bishop pine stands in Point Reyes that was started in the Bruns lab. And now still continue on. I think Kabir P at Stanford is still doing some research on that. But it is really interesting because, and honestly, the reason that Bishop pine survived so well in Humboldt County is because green diamond slash and burns when they do clear cuts. And so when they do that, it offers an opportunity for those seeds to germinate. Yeah.
Do you think there's going to be a study on the tree culture and tree survival after the Southern California fire?
I do follow some mycologists who in Altadena and the Altadena Hills did like a lot of research on, I think that's like a lot of oak, maybe some like pine, who had been studying, had study sites there. And I bet that a lot of people will build upon that research by studying what's going to be fruiting in the next few years, because that's a lot of high severity fire. And that can actually like ruin an ectomycorrhizal community.
We have two more here.
Okay.
Would you briefly outline the mycological studies program that exists these days at Cal Poly Humboldt? Who's heading it up?
Okay. Okay. So if you want to do your grad degree at Cal Poly Humboldt, which I highly recommend because Humboldt is such a beautiful place, you will be studying under mycologist Terry Hankel. And he is a very learned mycologist. He's done a lot of things in tropical mycology. He's a big shot head editor of Mycological Society of America. But he is, he will push you. And he is kind of the the mycology professor. So if you are interested in working with him, you should first reach out to me and we can talk. And then you should reach out to him. Because he has guided a lot of students through mycology, but is in no way the only route to go for mycology. There's an amazing program at Oregon State. There's a good program at Cal. There's a good program at Stanford. But if you want to come to Cal Poly Humboldt, you should definitely reach out to my email.
There's only one person?
There's only one person.
All right, next question. Does the lumber industry care about fungus? Could you get private funding based on the likelihood that optimum timber production, especially in replanted areas, depends on correct understanding of the issues that your research addresses?
I think that's an optimistic, like that would be optimistic that they would be interested in this research. I mean, you think because it would be like, okay, these are the associates that confer success, but I could see experimental forests like Refi up on the North Coast doing stuff like that. But with Green Diamond, that's just not really Green Diamond, Mendocino Redwood Company or Humboldt Redwood Company. That doesn't really seem to be their direction in terms of what I've sussed out from them. And they're more worried probably about finding rare fungi and then not being able to cut is what I would think. But I'm being maybe a little unfair. This is just when I worked with Sierra Pacific, for instance, like they made a crazy concoction of herbicide to suppress shrub growth, which is something you have to do after a fire if you don't want the next succeeding ecosystem to be shrubs. The best way is to spray a bunch of herbicide, but herbicide can also include things that are also fungicide. And so you'll get these plantations of pines that don't really look very healthy, but they're coming up because they have no competition. And there probably are still some ectomycorrhizal species in the soil, but maybe someday that would be a thing, but I can't speak to my confidence that that would be a thing soon.
Are all the ectomycorrhizal fungi that have fruiting bodies saprophytes?
Saprotrophs. Saprotrophs?
Saprophytes is what they wrote.
Okay, I'm gonna assume saprotrophs. Partially, they are able to decompose. They're able to basically exude enzymes to extract minerals from the soil and nutrients. But again, they're getting most of their carbon from the host tree and not from decomposing litter on the ground. But they are able to do to some extent, saprotrophic decomposition, but they're not evolved really for that to be their main source of carbon. All right. Okay.